Okay, students, this is going to be about viruses. This presentation will focus on the different kinds of viruses. We're going to learn about how they cause disease. We're going to learn how they enter the cell and what can be done to treat infectious diseases that are caused by viruses. There are different components of a virus that enable it to cause disease. We'll talk about that. And we'll learn about the different uh, basically strategies, if you will, that a virus employs in order to cause infection. And we will learn about ways that viral infections can be managed when there's a pandemic situation. So, viruses are quite different from bacteria. And we've learned, of course, that the bacteria are living cells. They carry out the same basic characteristics of life that the cells of your own body carry out. And of course, you have a lot of bacteria living inside of you, like in your gut, for example. But the bacteria are free living. That means that they can metabolize nutrients independently, they can reproduce independently, they can self regulate or maintain homeostasis independently. And they, of course, have very sophisticated ways of communicating and responding to their environment, whether it be other bacteria or maybe even the cells of the host that maybe they've infected, if we're thinking of a pathogenic bacterium. Viruses are a completely different infectious agent. Most virologists are in agreement that viruses are not living because they cannot carry out those processes that I mentioned earlier that are associated with living cells. They cannot carry those out independently. Now, viruses can reproduce, but they're utterly incapable of doing so independently. They actually need to utilize the intracellular machinery of the host that they enter. So they are called a intracellular parasite. They're very, very tiny. Most of them are about a thousandth the size of a bacterium. And of course, you've heard about vaccines. Vaccines, of course, usually consist of proteins that are associated with a certain virus or bacterium, or sometimes vaccines contain weakened forms of a virus or bacteria, uh, especially in the case of viruses. So we'll talk about antiviral treatments a little bit later, but viruses, of course, are very tiny, but they still have some components that we want to be familiar with. So there are nucleic acids that are inside the virus. I say acids as if all viruses have more than one nucleic acid particle. That's not the case at all. Some do, some don't. Like this virus that we're looking at only has one nucleic acid molecule. It's going to be DNA or RNA. There are viruses that have more than one. Um, the capsid in green is a protein coat or a shell that encases that nucleic acid. And then many viruses, especially the ones that cause disease in animals, have a viral envelope, which basically consists of the phospholipids that originated from the host cell. And then, along with those phospholipids, you have proteins on the surface. And those are really important for helping the virus gain entry into the host. So let's talk for a second here about viruses and how they get in. So here's a host cell membrane right here. And then here's a virus with its viral envelope. And there are proteins on the surface of that viral envelope that have a specific molecular configuration that enable them to fit like a key into a lock with the receptor on the surface of the host cell. Now, that receptor is probably used for other purposes. Maybe it's used for interacting with other cells in favorable ways that help a multicellular organism. But unfortunately, sometimes those receptors might also have a high affinity or chemical attraction to certain viral proteins. And so this protein is called a hemagglutinin. Anyway, the host cell is targeted by the virus, and a lot of viruses will cause disease. And so the common cold is caused by a virus. It's called the rhinovirus. And Of course, we've heard about coronaviruses, which target the respiratory tract. So viruses generally can reproduce very rapidly within their host, and as they reproduce, they also have the capacity to evolve. So they can mutate, and so RNA viruses particularly mutate very rapidly, and this is because they do not utilize um, the proofreading mechanisms that our cells use when they are copying their DNA and reproducing. So that means these RNA viruses can adapt very rapidly, and they can actually change the configuration of these proteins on the surface of the viral envelope. So, for example, this COVID virus that you guys have been hearing about, it's called SARS-CoV-2. It was, they think, possibly originated in bats or some other kind of animal, maybe somewhere in China. They're not totally sure. But the disease or the symptoms that we've read about in the news in humans might not have necessarily appeared in bats or whatever animal vector that virus was being harbored in. But when those animals come in contact with humans, well, now the virus has a way of jumping from one host to another. But maybe now, with that transmittance from one species to another, if that virus has proteins that are specific for the receptors that are on the surface of, let's say, the cells of the respiratory tract or in the lungs, well, now we have a virus that can cause disease in humans. So, viruses come in all different shapes. You can see them here: cylindrical, icosahedral, right here, spherical. This guy right here to the far right causes disease, or I should say causes harm to bacteria. This is what they call a bacteriophage. It looks rather robotic, rather geometric in shape. Definitely a virus. You see the shaft here through which the viral DNA can be injected into the host cell. And I'll show you how that works in just a minute. So, viruses of all different kinds have affected humans. We've learned about the coronavirus in news, and I just told you a little bit about it. The coronavirus is an RNA virus. It is an RNA virus, so it'll mutate very fast. Smallpox virus wreaked havoc on humanity for quite a few times, quite a few episodes in human history. A vaccine was developed in the late 70s for that. Pretty dreadful disease, causes lesions all over the body, and has caused a lot of death as well. You've heard of herpes, hepatitis, human papillovirus, which is a sexually transmitted disease, and then chicken pox, smallpox. These are DNA viruses. But some of the ones we've heard about in the news lately, like the flu and the coronavirus, those are RNA viruses. HIV, it's an RNA virus. And some of these others that you've probably been vaccinated against, like polio, measles, mumps, these are RNA viruses as well. So what happens when a host cell gets infected? How does the infection actually happen? Well, here we've got a virus, makes contact with the host cell in the way I described earlier. The proteins in the viral envelope fit rather nicely with the host cell receptors if infection is going to be caused. And then if this is a DNA virus, the DNA gets released into the cell, and that'll get transcribed into RNA. Now, wait a second. How does that happen? Well, the host cell enzymes oftentimes are involved in this process. And then, of course, the host cell ribosomes will take the RNA and then translate those into proteins. Those are viral proteins. And of course, other enzymes, sometimes it's enzymes in the cell, but it might be enzymes from the virus that will make copies of the nucleic acid from the virus. And so we have many copies of viral DNA here. And then the virus assembles itself in the cell, and then it goes on to infect other cells. So here you'll notice that the lytic cycle is a cycle where cells typically will die or they will be harmed significantly. And so the effects of the infection are observed very soon after the virus enters the host. But with something called the lysogenic cycle, the symptoms may not necessarily surface right away. The virus has more of an undercover operation that it employs. And so the DNA of the virus gets integrated into the host cell DNA. And so here's a virus here. This is the bacteriophage. There's the nucleic acid in the head of the virus. It gets injected into the host cell. And you can see that here's the host cell DNA. The viral DNA enters into the host, and you'll notice that DNA is transcribed into RNA by the host cell machinery, like RNA polymerase. And then ribosomes will take the RNA, translate that to make viral proteins. Viruses are assembled, they're released to cause disease. And so this is what they call the lytic cycle. The cell often is destroyed by the end of this process. On the other hand, there's a lysogenic cycle, and that involves an integration of the viral DNA in this case. You'll notice when we go from the left to the right, we have the host cell DNA, the viral DNA, but you'll notice if you go further to the right here, viral DNA is incorporated, it's integrated, or it's incorporated into the host cell DNA. And so that means when the cell reproduces, and when the cell copies its DNA, it's also copying viral DNA. So even though the cell itself is maybe not affected negatively by the virus at this point yet, it is kind of doing a service to the virus by copying the virus or viral DNA. And then for reasons that are not well understood, there's a transition from the lytic cycle to the lysogenic cycle. If you go from right to left down here, you'll see that the virus becomes activated, and the cell's host cell machinery essentially becomes hijacked and is forced into a state of slave labor to make copies of the virus. And the viruses will go on to invade other cells once this lytic cycle comes to a completion. So there are other viruses that will infect in a slightly different way. On the surface, mechanically, the same happens. You have the receptors, but once the virus enters, the viral genome gets reverse transcribed, I should say, into viral DNA. So this is actually an RNA virus. So, for example, let's say this is a COVID virus, SARS-CoV-2, and it enters the host cell. And then the viral RNA gets copied. And actually, the COVID virus replicates a little differently than the way this is shown here. But I am correct when I say that the COVID virus is an RNA virus. But many viruses like this will then have their mRNA basically used to make copies of viral proteins. So that's the translation step there. And so once you have enough copies of the virus, then the virus will then fuse with the cell membrane, and then it will be released to the outside of the cell. And so that's how an RNA virus typically causes disease. All right, so the retrovirus, on the other hand, is a little more complicated. Again, these are RNA viruses. I should go back to this. They use an enzyme called reverse transcriptase. And once their genome is converted into DNA, it's then inserted into the host cell DNA. And then at that point, we call it a provirus. All right, so how does this work? Well, like with HIV, the RNA enters the host cell once the receptors and the viral proteins lock together. The nucleic acid enters the host cell. That viral RNA is reverse transcribed from RNA to DNA. Now, you might remember that in eukaryotic cells and in bacteria, the viral DNA, not viral, but cellular DNA is transcribed into cellular RNA. But in this case, it's the reverse. The viral RNA is reverse transcribed into DNA. So there's a special enzyme called reverse transcriptase that's originally contained in this virus. Once we have viral DNA, that gets incorporated into the host cell genome, and it embeds itself. And so the cell will then go through many cycles of reproduction. It'll replicate DNA, its own DNA, but it's also replicating the DNA of the virus. And it may go through many, many, many life cycles, many cycles of reproduction. In the case of HIV, this goes on for years. So someone may not really be symptomatic for years, and then for reasons that are not totally understood, the virus activates. And so then the virus will basically make copies of itself. But again, it uses the host cell machinery to do this. So the viral DNA gets transcribed into viral RNA. That gets translated into viral proteins. The proteins are assembled to make viruses, and the HIV will then leave the host cell. All right, so here's another picture of HIV. It attaches at the surface. It then enters the host cell. Um, well, this is a virus actually leaving the host cell to the right, and so it fuses with the cell membrane, and then after that, it actually kind of coats itself in the phospholipids that make up the cell membrane, and that would constitute the viral envelope. And then it goes on to infect other cells. So HIV causes AIDS. AIDS stands for acquired immunodeficiency syndrome. And so when someone's white blood cell count drops to a certain level, one of the things that that patient might be tested for is HIV because it attacks the immune system. The host cells that the HIV targets are specific white blood cells called T helper cells, and they help the immune system with communicating with other types of immune cells so that infections can be eradicated. But when someone's own immune system is the target of the virus, it's very serious because if their immune system is wrecked, then they become sick from a large plethora of different bacterial or viral agents. Okay, so smallpox, herpes, I just mentioned those. These are caused by viruses. Pandemics, of course, are infections that occurred on a much larger scale than, say, in just a family or in a neighborhood. If we're talking about across many communities or across the globe, we call it a pandemic. 1918 was the worst pandemic in several centuries, and so there were about, I think, 700,000 people that lost their lives just in the United States alone. The COVID-19 disease that's been making the news has racked up a significant death toll, but still nothing in comparison to the 1918 pandemic. And it's difficult to determine how you classify these deaths. Did someone die with the virus or of the virus? Because some people might have underlying conditions that predispose them to be affected by a certain viral infection. But anyway, so vaccines, basically, they're made up of weakened viruses or viral proteins that are similar enough to the actual virus that it can alert the immune system and stimulate an immune system to produce special proteins called antibodies, which will then tag viruses that you might come in contact with at a later time. And your immune system, of course, having all those antibodies, can very, very rapidly mount an immune response against that virus. So if let's say you got the polio vaccine, and let's say in a few years' time you come across the polio virus, you get infected with it. Well, your immune system already has all the antibodies that are specific for that polio virus, and it can quickly tag those polio viruses, and then you have special cells in your immune system that will target and then destroy those viruses. And so your immune system gains the upper hand very, very quickly if you've already been vaccinated against that particular virus. If you've been exposed to a certain viral infection, then that's another way to gain immunity as well. It's not often as fun. Sometimes you get sick in the process of becoming immune to a certain disease, a certain viral infection. So, you know what? I forgot to mention earlier vaccines. When I think of vaccines, I think of Jonas Salk, for example. He developed the polio vaccine. And I shared with you a video earlier of how he invented this vaccine and he used formaldehyde to weaken the polio virus. In other words, he wanted the vaccine itself to have a form of the virus within it that wouldn't be able to cause disease, but was chemically similar enough to that virus that it could elicit an immune response when someone was injected with that polio vaccine. So it's a marvel of medical innovation. And Jonas Salk is a hero. He saved hundreds of millions of lives if you count all the decades afterwards when many, many, many people were vaccinated. And polio is a terrible disease. I mean, people either died when they got it or they got because it attacks the nervous system. Some of them ended up in what's called the iron lung, which is a pressurized chamber that helped the person breathe, because if they were paralyzed from their neck down, they couldn't operate their diaphragm to breathe. So it was a terrible illness, polio was. All right, so can you use antibiotics against viral infections? The answer is no, because the antibiotics that we use are targeting the ability of bacteria to reproduce and build their cell walls. And viruses are not structured like bacteria. They don't employ the same metabolic mechanisms for maintaining their cellular hardware. They don't have cellular hardware, of course, but bacteria do. So antibiotics don't work. And so the antiviral drugs that maybe you've heard of, like AZT, for example, it attacks the HIV virus. How does it do that? Well, it actually interferes with the virus's ability to reproduce. The reverse transcriptase enzyme that HIV uses to reproduce is the target of that particular drug. And you may wonder, well, why is it that if you got, let's say, the common cold, you go to the doctor and the doctor gives you an antibiotic, why would he or she do that if it's not really effective? Well, sometimes when you get a viral infection, like in your lungs or in your sinuses, the immune response causes a large production of mucus. And there's a lot of inflammation. You got a lot of cells sloughing off and dying from the infection, from the viral infection. And so basically, your sinuses become like quicksand. And so then there's what's called an opportunistic infection. So bacteria may come into your system, and then you might end up with a bacterial infection. So, for example, pneumonia could easily follow after a respiratory infection. That's why you hear a lot about older folks or immunologically compromised people. They get the COVID-19 when the virus damages lung tissue. And the immune system is generating all that mucus, then bacteria can easily get in, settle, reproduce, and then pneumonia follows after that. And oftentimes these folks, if they're immunologically compromised, they end up dying of pneumonia, or you could say they died of COVID-19 in an indirect sense. All right, so that's a little bit about viruses, and I hope that was helpful.